with any evidence to support it. And, if it helps, because there have been some newer papers and people have been sending these around about that, saying that the genome has been sequenced for this new virus. So, if you want me to talk about that, there's no such thing has been achieved. It's a total farce, but I can explain how it works if you like.
Yes, please. Yes, please. Just to finish off what you said before, because I felt it was very important. So I've listened to your other talks. When you said they couldn't prove anything, remind me what it was, because it's all the drugs I've done in my life. It's like I forget things. But there was that guy, Torsten Engelbrecht, wasn't there, who actually asked them. So tie those two in just before you move on to that thing there, please.
Yeah, well, that's a really important piece of journalism. So it's in Off Guardian, and it's something like "The COVID Test is Scientifically Meaningless" is the title, by Torsten Engelbrecht, and he's also the author of the excellent book Virus Mania that I encourage people to check out. But in the article, and he co-wrote it, but I can't remember his co-author's name, so I'm sorry not for giving you credit. But what they did was they actually corresponded with the authors of all four of those papers and asked them directly, did you purify a virus? Because the only way to prove that a virus exists and to characterize it, like to see what it's made of, what its structure is, what its DNA or RNA sequence is to purify it from a person with an illness, right? If you're trying to show that it causes an illness. So you purify it out, and then you can actually look at it, right? Like, imagine if you wanted to figure out like the shape of a human body, if you had, every animal species in the world mixed up in a test tube, right? And just pulled out random samples and tried to reconstruct a human from it. You wouldn't be able to do it. You'd get some kind of chimeric theoretical model. You'd have to take a human out and just humans, and just look at humans only, separate them out from all other animals, right? And then you could say, okay, they have 2 arms, 2 legs, 2 eyes, right? All these things. So it's the same thing with a virus. If you can separate it and purify it and say, okay, I just have this virus now, I can look at it, it's spherical, it has, proteins on the surface, it has proteins inside. That's the only way you can look at it. But that's not what's been done in any of these experiments. And the authors of all those papers directly admitted it in their correspondence, that they did not purify any virus in any of these experiments whatsoever. And this ties right into the genome, because you have some newer papers coming out, and people have been using them to challenge some of my assertions. And saying, oh, look, we have the sequence of this virus. Like, if we have the sequence, then it must exist. But the thing is that they don't actually have the sequence. What they have is a theoretical computer model of a genome sequence. And so, as I said before, right, if you wanted to characterize the structure and the contents of a virus, you purify it out. Then when you have it by itself, you could pull out the RNA that's inside of the sacs, and you can use an enzyme to digest the membrane and release the RNA. Then you have an intact piece of RNA that represents the whole genome of the virus, if it exists, and you could sequence that from end to end, right? And that's how you would determine the sequence. If what they've actually done is they took a dirty sample of lung fluid or from a nasal swab that has lots of different sources of genetic material, right? Human cells, bacteria cells, fungal cells, all kinds of things. And then they use probes for PCR that they've pre-identified as being viral from a computer database, right, that they've created from other studies they've done the exact same way, right? And they say, well, we know these are viral sequences, so we look for these. And then they amplify a library of short sequences about 200 to 300 bases long, where they say the entire genome is 30 or 40,000 bases long. And in one study I looked at, they actually had 20,000 different fragments, short fragments, that they amplified. And then what they do is they put these into a computer modeling software that's connected with the database of those other virus sequences that were all obtained the same way. And they let the computer program piece these little puzzle pieces together into one long strand. And so, you know, it looks where sequences overlap and tries to puzzle them together. But then, of course, there are gaps. And so what it does is it just fills in the gaps from that database with imaginary sequences. So we basically have this kind of like Frankenstein approach to a theoretical computer model of a genome of a theoretical virus that's never been purified. And that's what they're saying is the genome.
Hey, but Dr. Kaufman, that'll do. Hey, it's like, you know, we came from apes. Ooh, 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 you know, that'll do. We don't know. So, look, I'm not saying I know where we came from. I'm not talking about God or anything like that. I'm just saying, isn't it just magnificent? And we don't know. We haven't got a clue. But what they'll do is they'll use these things to manipulate humanity with fear. It's the same thing with the religions and that. It's like, well, that'll do. Hey, we can get them to do this, and we'll punish you if you're not a good girl and boy. It's like, stop it now. Good grief.
And Jason, it's not that they do this because the scientific procedures to do it the right way are too difficult. Because when they do exosome research, which is looking at the same size particles with the same characteristics, just they're from our own cells, right? Probably what really viruses all are. They can do that no problem. They can purify out the exosome and they can sequence the genetic material right out of it. So it's not that they can't do it. It's that they know if they try to do it that way, they're not going to find anything. It's going to be because there is no virus. And so they don't do it that way because of that, they won't get a paper published.
And then they won't control humanity, which I know that we haven't got long because this goes quick with you. I could speak for 10 hours. But so in a quick loop back to where I said those people were saying to me, but I've been really ill and bless you, I love you so much if you're watching, yeah? But you might have had pneumonia or something, some bad pneumonia, you know, or something like that. But some of them have said to me, but I had the test, it was COVID. Yeah, well, so is a pawpaw and a goat, right? And lots of other people, right? It's like, holy crap. Hey,
it's hard to accept that the test is completely meaningless. Like, you know, people want to say that it has an 80% false positive rate because there was one retracted study where they you can't measure the accuracy rate because there's nothing to compare it to, right? And there's nothing to definitively identify this illness to compare it to. So, but there was this one study, they tried to estimate it. They also used mathematical models, and they said it was 80% false positive rate. And a lot of people are throwing that number around, but I disagree. I think that the false positive rate is 100%, because there is nothing tying this test to an illness that you're trying to measure. And since they're totally disconnected, then every result is false. So it's 100% false positive, 100% false negative, right? It's the error rate is 100% for this test. It's completely meaningless. And if you just look at a few documents, you should be able to figure this out because it's essentially written by the government themselves.
And sorry, yeah, Dr. Kaufman,